All right. Well, good afternoon, everyone, at least EST time. Welcome to today's seminar series. Um, we're excited to have you. I just wanted, before we get started and introduce the speaker, I want to remind everyone to please renew your annual memberships. It's not a lot. It's free for trainees, but it's $100 for professors. But it's very important that you renew it because we need to maintain membership, and that's gonna, strengthen our collaboration within the functional precision medicine community. So please, 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 I urge you to just renew your memberships if you haven't today. Without further ado, it's my pleasure to introduce today's seminar speaker, Dr. Rita Fior, a developmental biologist and group leader at the Champalimaud Center for the Unknown in Lisbon, Portugal. Dr. Fior leads the Fior Lab, where her research focuses on cancer development, personalized medicine, and innate immune evasion. She's a pioneer in the development of the Z Avatar model, an innovative zebrafish-based platform used to predict patient-specific responses to cancer therapies. This work has garnered widespread recognition, including features in Science, Nature Reviews, National Geographic, and The Independent. Dr. Fior is currently spearheading a randomized clinical trial to bring this approach closer to clinical application. In parallel, her lab is uncovering new mechanisms of innate immune evasion by studying interactions between human tumor cells and the zebrafish immune system. Dr. Fior's work has earned numerous awards, including the CRI Impact Award in 2023, and she's also a co-author and editor of the textbook Molecular and Cell Biology of Cancer. So please join me in welcoming Dr. Rita Fior, who will be speaking today on zebrafish avatars for personalized medicine and discovery of innate immune mechanisms. Just a quick reminder that if you have any questions during the talk, just add it in the chat, and then at the end, we'll open the floor for live questions. Just raise your hand. All right, thank you so much. Welcome, Rita.
Thank you. Thank you so much for this opportunity to talk about our work here at the Champalimaud Foundation and to the Society of Precision Medicine. Thank you so much. So I always start my talk with this slide, which for this audience, I don't think we need it so much, but it's just to remind us that we are surrounded by amazing technology. We can go to the moon, we can fly planes every day, and we have amazing technology in biology. But still, we don't have a functional test to, like an antibiogram in the clinic to help optimize and tailor treatments. And I think we all here in this society are really thriving to get there. And this was really my driving force to start developing the Zebrafish Avatars for personalized medicine, which are basically patient-derived xenografts where we take cancer cells from patients and inject them in little zebrafish embryos of 2 days post-fertilization, generating these avatars from the patients. We take tumor cells from surgery, from biopsies, or from fluids that have cancer cells. And then the next day, we have a group of avatars that are control and the different treatments, which can be chemotherapy, biological therapies like antibodies, small molecules, or even radiotherapy. So we developed several protocols for all these therapies. And here on the left, we can show in this small video how we microinject these cancer cells that we label them with a fluorescent dye. You can see here the eye and the heart of the fish, and we microinject. So we really reduce the scale of the cells of the tumors, and that's why we with a small sample, we can generate many avatars because we're reducing the scale, and then we reduce the time because we can take all the whole fish into a microscope and image them. So our big goal is really, like, I guess everyone here in the society, to really help MDs choose the best option of treatment for their patients. And so I'm just going to give you a roadmap and what we do in the lab. So we use transgenic zebrafish animals that, for instance, can have their vessels labeled with GFP, so we can look at angiogenesis, I'll show you an example, or other reporters from the host. And then what we do is immunofluorescence for the tumor, and we can zoom in into this tumor by confocal imaging and really have single cell resolution of the whole tumor. Okay, so we had to show that human cancer cells can actually proliferate in the fish. This is a 2-hour EDU pulse in the water where the fish are swimming, and you can see that cells are undergoing S phase, a Ki67 staining. And here, for instance, is an example of a triple negative breast cancer, human breast cancer, that is able to recruit blood vessels from the fish. So it's highly conserved, the molecules between human and zebrafish, and that's why you can see that the human cancer cells can actually recruit blood vessels from the zebrafish. But a big advantage of the model is that it is an in vivo model, and we can look at metastasis. And here's an example of a micrometastasis in the eye of the fish. The other advantage is that we can take the whole fish live into a microscope. And we can study biology and study how cancer cells, for instance, migrate and metastasize, but we can also study how cancer cells interact with innate immunity. So this is a transgenic fish that has labeled the macrophages in green, and they're just clearing this tumor. And I'll show you in the end of the talk this part of the work of the lab that is really dedicated to trying to understand these interactions. Okay, so as a readout of our assay, we look at activated caspase-3 to look at cytotoxicity, basically, by microscopy, by confocals, or spinning this microscopy where we can scan the whole tumor. So we always have a group of controls, fish, in this case, like 33 xenografts, and then another group for the drug A or drug B, in this case, Folfox and Folfidi. And we quantify the apoptotic bodies in each tumor, and each dot here, as you see, is a xenograph. So this gives us numbers. So this is our roadmap of what we do in the lab. So these last years, yes, it's a beautiful model. We can have beautiful images, but is it predictive of patient response? That was really my big goal in these last years, was to really test the predictive value of the model. And for that, we developed several co-clinical studies where basically we generated avatars and treated them exactly with the same treatment as the patient, to see if the avatars could actually predict whether the patient was going to respond or not to treatment. And today I'm going to show you in more detail the study in colorectal cancer that was recently published. So we had 55 patients of colorectal cancer that went through surgery, and after surgery they got therapy X. It doesn't matter, there is several possibilities. And then with the surgery sample, we generated the avatars. And we have a group of controls and a group of therapy X. And the question is, can the avatars actually predict whether that patient is going to respond or not to the treatment? And so here is a plot of the 55 patients where we are plotting the average fold change of the avatars. So how much did the drug induce apoptosis in the avatars, the drug that the patient was treated? And as you can see here, stable patients that were stable, that did not progress, their avatars have a much higher fold change of apoptosis than patients that actually progressed. And this was statistically significant. We did then, a ROC curve to define what could be the threshold to define sensitivity and resistance to the test. And then we did the predictive table or a confusion table. And as you can see here, so most of the patients that did not progress, their avatars were actually sensitive to the therapy, whereas most of the patients that actually progressed, their avatars were resistant to the therapy. And so we got really, I think, really good predictive values, positive predictive value of 92% and a negative predictive value of 71%, which are already really pretty good values. But the Zebrafish Avatar model can give us more information because we are in an in vivo context and we can look, for instance, into metastatic potential, okay? So the formation of micrometastases in the fish. So we inject cancer cells, and some patients, some cancer cells don't do any micrometastasis. We cannot see the formation of this. But some others, you can see a lot of micrometastases spreading and disseminating through the fish. But this micrometastasis potential is not an evolutionary potential. It's just whether cancer cells are able to form, to go into circulation, and to resist shear stress in circulation inside the fish, whether these cancer cells are able to evade innate immunity, because at this stage of development in the model, we only have innate immunity. So, but cancer cells, to actually form these micrometastases, they need to evade innate immunity, otherwise they'll get cleared, which is what happens here in this fish. So what we saw was that early stage patients, most of the avatars from early stage patients did not form micrometastases. However, late stage 4 metastatic patients, they do form metastases. This was expected. Also, patients that progress, they form more micrometastases. This was also expected, but it was good to see. Well, at this point, this was not really black and white, and we didn't know more what could we do with all our data. So we did a multivariate analysis with all the data that we gathered from the patient, around 15 variables, whether this is left, right, rectum, if the cancer had perineural invasion, staging, etc., sex of the patient, and also the variables from the avatar model, okay, that we had around 5 readouts from our assay. And this multivariate analysis, what gave us was that there were only 3 variables that were predictive, had some predictive value on the outcome of the patient. One was staging from the patient. Early stage, late stage, better prognosis if they're early stage. But 2 from the avatar. One apoptosis that we already know was highly predictive, but other metastasis. And this allowed us this analysis to develop a 3-decision model, okay? So this 3-decision model told us that we should separate patients in stages, early stage and late stage. And then the model asks us in early stage, the first thing the model asks is whether the cancer cells that came from the patient, from the surgery, if they have metastatic potential or not. If these cancer cells are not able to form micrometastases in the fish, the model predicts that the patient will not progress, and the model doesn't care about chemotherapy. However, if these cancer cells do have metastatic potential, so they form micrometastases in the fish, then the model asks about chemotherapy, whether these tumor cells were sensitive or not to the therapy. In stage 4 patients, the model doesn't care about metastases. These are already metastatic patients that have metastases, and it follows apoptosis values. So now if we plot in a prediction table using this model, so now we really increase the negative predictive value. So we have a positive predictive value of 91% and a negative predictive value of 90%. So this is pretty high and really, I think, outstanding. But what I think is really cool is that this model is taking into account not only the cytotoxicity, of course, but also the biology of the cancer cells that we are being able to capture in our model. And I think it was really cool to have this in a very unbiased manner. Okay, so then what we did was to plot the progression-free survival of the patients according to their avatar test, whether it was sensitive or resistant according to the 3-decision model. And I hope you can see there's a huge difference, but actually showing that if you give the right treatment to the right patient, this makes a huge difference. And I think that's why we are all trying to develop models for personalized medicine. Okay, so over these years, we've been really trying hard to show scientifically validate the model and trying to get the predictive value. And so I showed you the colorectal cancer story, but we have a very similar study in breast cancer and ovarian cancer. In breast, we had 100% predictiveness, so it's probably a question of numbers, but in ovarian cancer, we have around 90% as well. So we are now very confident on the predictive value of the model. And so we now moved on and just opened a clinical trial, a randomized trial. And I have here a small video. I don't know if you're going to hear the sound that explains the trial. So I'm going to try to make it happen. Can you hear it?
We have designed a clinical trial in which breast and ovarian cancer patients will be randomized into two groups. In the first group, patients will be treated according to standard of care as decided by an oncologist. In the second, tumor cells will be collected and implanted into Zebrafish Avatars to find the optimal treatment for each patient. The only difference between the two groups will be whether the treatment is chosen by the oncologist or by the Zebrafish Avatar test. Based on our preliminary studies, we estimate that the Zebrafish Avatar test will double the overall survival of this group, despite their advanced stage of disease. This would not only buy patients more time, but also improve their quality of life and spare them the toxicity of ineffective treatments, as well as decreasing healthcare costs and paving the way to treat other cancer types. We believe that a truly personalized test will optimize the effectiveness of all currently approved therapies, which could have a tremendous impact on the standard of care and overall survival of cancer patients worldwide. But to bring this groundbreaking Zebrafish Avatar test to the clinic and help doctors choose the best treatment, we need your help so we can get the right treatment to the right patient and usher personalized medicine from the future into the present.
So it's a multicentric trial. We just opened and we are waiting for the first patients. So we are going to focus on metastatic breast cancer and advanced ovarian cancer patients that have fluids, malignant pleural effusions and ascites, because these fluids have to be drained from the patient to alleviate symptoms, but then they're trashed, so it's medical waste. And we found out that these fluids are very rich in cancer cells, and we can generate hundreds of avatars with this medical waste. So this allows us to do a low-cost trial or lower cost trial, but also avoid ethical issues. So we don't need to perform any additional procedures in the patient to get the samples. And so these patients, the eligible patients, will be ones that have fluids, but also have more than one choice of treatment, okay, which is second line for ovarian cancer and third line for and onwards in breast cancer. So the trial is designed that is the oncology in an appointment that actually chooses which are the options of treatment that are available for that patient. So within the guidelines, the international guidelines. So it's not off-label. It's for the currently approved therapies and that are being given every day in all the hospitals, okay? So it is the oncologist that has to go into the ECRF and put in which are the options for that patient. And only after that, he can randomize the patient towards or oncology choice or to the avatar. And then if it's within the oncology choice, he has to choose one of the options that he or she put in to the ECRF. If it comes to the Zebrafish test, we will test exactly the same options that were defined previously and then see which one will give a higher cytotoxic effect in the avatars. And so our goal is really to compare PFS, OS, and response rate of these two groups of patients. Okay, so this is going on, just started, so it's really exciting for the lab, and it's been a real difficulties to get it approved and all the regulations and so on, but it's done and it's open now in these 6 hospitals at the moment. And we're waiting for the first patients to come in. So I'm just going to wrap up this part of the personalized medicine and then going just a teaser on the innate immune part. So I think we also discussed a bit in the society how sometimes, not many times, doctors come to us to help with when we can have this model that can actually help them choose therapies. But we do have a case study where in glioblastoma, where an oncologist came to us. She had a patient, very young patient with gliosarcoma, with a rare type of glioblastoma, which is gliosarcoma. And the patient went to his second relapse and third surgery. And at this point, the tumor had been sequenced. There was no actionable mutation. So she had around 13 options of treatment of chemotherapy to combine with radiotherapy to see if they can, give more time to this patient. And so we tested the 13 options. So Marcia in the lab tested them. And when we do glioblastoma brain tumors, we inject in the hindbrain of the fish instead of the perivitreal space, which is the normal place where we do it. And so we did the 13 options, and what Marcia saw was that combination of radiotherapy with pemetrexed and pemetrexed with doxorubicin could be better options than the other ones. And so this patient went to surgery, decompression of the brain. He was better from his symptoms, but 20 days after surgery, these symptoms came back again. So it was highly aggressive, this tumor. After that, started the therapy that we found that was more efficient in the Zebrafish Avatars. And the patient recovered from his symptoms after therapy, and actually was stable for around 2 months, which is not much, but this is a very aggressive tumor. And I think we came quite late in the story of this patient. And so hopefully in the future, we start earlier trying to help tailor the treatments as the patient progresses in a more efficient way. Okay, so this was the personalized part of my lab. The other arm of my lab is really trying to study the interactions between tumor cells and innate immunity. And why did I start interested in this? Because when we started this work, I realized that some cancer cells, human cancer cells, implant really well in the fish. So it just is a very short assay, just 4 days. So we inject the cells and we stop the assay 4 days later, sometimes 3 days. So it's really fast. And these 4 days, we see the tumors, it's fine. But some other human cancer cells we inject, and after 4 days, they disappear from the fish. So we don't see any more cells. So as I told you before, at this stage of development, we have innate immunity. But it could be just that human cancer cells were not happy in the fish. So we had these 2 hypotheses. But the fact is, when we image live what's going on in the fish, so this is the dorsal aorta. So in red, you have the cancer cells, and in green, you have myeloid cells. What it seems to be happening is that myeloid cells are really chasing the cancer cells, the red cells, and myeloid cells are really phagocyting many cancer cells, as you can see here. But some other cancer cells are invisible to these immune cells. So this raised the hypothesis that maybe these regressor tumors are being recognized by innate immunity, and the progressor tumors were able to block this reaction. So this is published, so I'm going to be fast here just to show you that we actually showed that this is happening in the fish. So we did a study where we focused on 2 cancer cell lines, colorectal cancer, that one is from the primary tumor and the other from the metastatic site from a lymph node metastasis, that had these completely opposing phenotypes, but they came from the same patient. So this was really an opportunity to try to understand what's going on. And so it could be just the primary tumor was not able to survive in the fish and had nothing to do with innate immunity. So what we did was so as you can see, for 80, sometimes you have a few tumors, small tumors, but 620 forms these really beautiful tumors in the fish. And our question was, if we mix them together, can 620 dominate over 40? Can it protect 480s from being rejected? So we did these experiments where you can play with the dyes and then mix them together. And what we saw was that indeed 480s, they do not implant really well, but 620s do. When we mix them together, 480s now implant much better, but the 620s goes down. Then what we try to do is try to see how is the tumor microenvironment composition. And what we saw was that 480 cells, the one that gets rejected, have much more neutrophils and macrophages in their tumors than the 620s and then the mix. The mix is really behaving like the 620, suggesting that 620 cells are really dominant over the tumor microenvironment. Then in zebrafish, we have tools to look at macrophage polarization using transgenic animals that label macrophages in red, but then we also have the TNFA GFP reporter. And so this really allows us to identify TNFA positive macrophages, and you can see that 480s really recruits more TNFA positive macrophages than 620s or the mix. And then to actually prove that innate immunity is really regulating this engraftment phenotype, we did a genetic and chemical reduction of myeloid cells. So these are hypomorph mutants. RUNX has low neutrophils, CFSR1 has low macrophages, and you can see when you reduce these myeloid populations, it increases the implantation of these cancer cells. Also, if we use L-cladronate, now 480s implant almost 100%. L-cladronate really abrogates completely all macrophages from the fish, just like it's done in the mouse. Okay, so finally, we also did an innate immunoediting experiment in zebrafish, where we injected 480 parental cells into the fish. And then, so at 1 day post-injection, we have all fish with a tumor, but then at 4 days, 80% of the fish, the tumors disappear. But the fact is that 20% of fish do have tumors. So we dissected these tumors and injected them again. So these tumors seem to be escapers. Can they block this innate immune reaction? That was the question. And so we re-injected these cells, and what we saw was that indeed these escapers can form much bigger tumors, but also they implant much better. Okay, so then reviewers wanted to know about signaling pathways and genetics. And so what we did was not really to sequence the 480 escapers, because these cells have been in culture. So what we did was to repeat the experiment and do a single cell sort-seq. So what we did was to re-inject again the parental cells. 1 day post-injection, we dissect the tumors and do sort-seq, single cell sort-seq. And then at 4 days, we dissect the escapers. And I hope you can appreciate here that we can see a really dynamics of the clones, the selection of the clones inside the fish. So what we see is the reduction of 2 big clones and expansion of other 2 clones. And basically the biggest clones that change, that reduce, are clearly clones that have not signaling on, but also interferon signature. And the clones that get expanded they have IL-10 signaling signature and Wnt signaling, and we're looking into this. But it was really cool to see these dynamics in the fish, so showing that we can use this model also to study innate immunity and interactions between cancer cells and innate immune system in the fish. Okay, so now we are really exploring this capacity of the zebrafish model to give us a lot of information about these interactions with innate immune cells. So this is a light chip movie showing macrophages clearing cancer cells. And I'm just going to finish with a small vignette of a screen that we did, the library screen, a phenotypic drug screen using these progressor zebrafish xenografts. So we use these xenografts that implant really well and then screen for drugs that could actually modulate the immune system to reject these cancer cells. Okay, so we just score the number of fish that have a tumor and don't have a tumor, and we got around 20 drug hits, and we followed up. And I'm going to show you just one example of one that we followed up, and we went to do experiment in mouse xenografts, because our goal here was to find drugs that could modulate the immune system, the innate immune system, to reject tumors or to switch the tumor microenvironment from a suppressive tumor microenvironment to a more reactive tumor microenvironment. So the idea is that maybe if we can combine these drugs that we can find in the fish together with immune checkpoint blockers, we can boost the efficacy of the immune checkpoint blockers. And that's what we did in the mice. And this is our preliminary data in the mice, but it was really cool to see this translation into the fish, into the mouse from the fish, where we see that this inhibitor of the muscarinic receptor can actually synergize with the anti-PD-1 antibody in mouse xenografts. And so now we're pursuing this, trying to understand the mechanism of action of this molecule in the context of cancer and the synergism with checkpoint inhibitors. We already saw that this drug is able to polarize macrophages, but we're still studying the real mechanism. Okay, so I'm just going to finish, I hope in time. I want to thank a lot my team. I've been really lucky with my team and former members, students, collaborators at the Champalimaud Foundation, collaborators in other hospitals, scientific collaborators, clinical, the platforms, mentors, patients, families, reagents from other labs, funding, and thank you for listening.
Thank you, Rita. That was amazing. Really incredible talk. And we met last year, and I was fascinated by this model. I'm going to start off with a question, and then if anybody has any questions, they can follow. But I wanted to ask you a little bit more about the number of drugs that can be tested using your model. And then let's start with that first. I mean, is there like a limit to the number of drugs you can test?
Yes, there is a limit. It's limited by the sample. So
Okay.
if we have a
So
lot of sample, we can test a lot of drugs. If we have few sample, we don't. So
it's really limited by the sample, not the zebrafish, or the number of
no,
zebrafish.
it's really the sample, because we do not amplify the sample in culture. So we get the sample from the patients, we cryopreserve it, and then in the day of the assay, of the injection, we thaw this sample, we prepare it for injection, and we inject. So we do not pass these cells through culture. So we don't really amplify them.
Do you dissociate, or you as is, you put the
No, we dissociate a bit, but not too much. So it's so that you don't get single cells, and we microinject together the tumor microenvironment of the patient into the fish. Okay, so everything goes in. But then, of course, if we don't have much sample, we cannot do many drugs because it is really our limitation is the amount of sample.
I hear you. And when it comes to your randomized clinical trial, the one you're just starting on metastatic breast cancer and ovarian, so the way you've designed it is very interesting, and I think that's the best way to be able to overcome some of the physician hesitancy, right, in using drugs that are off-label and all the challenges that come with it. So my question is, the way you've designed it is really you're going to test NCCN guidelines, but
Yeah.
you're going to tell the physician which of these are going to work best. That's the way you've designed it.
Yeah, that's the way I designed it. So the idea was, let's start by optimizing what we have now, because we do have a lot of tools and a lot of drugs already approved, right? And many times patients go through rounds of trial and error to find really the treatment that is more effective. But if you can at least increase the probability of getting the first one right within what we have. And of course, then in a later stage, for instance, in ovarian cancer, I think we'll have a big problem, which will be the multi-resistant tumors.
Yeah, and you don't have a lot of options too for ovarian as much as you do have for breast, right?
It's true, it's true, it's true. But we still, well, in the guidelines do have quite a lot, so we can combinations of chemo and stuff like that. So in the end, sometimes we have like 6 to 7 options that
Mhm.
were that are in the guidelines, but really are chemo based, right? It's not, you don't have so many targeted therapies or whatever. And the multi-resistant tumors will be a problem, which I would like to solve, but then I need to think how to design a parallel trial to go off label in these patients that really need to go off label. But I think here there's also a space for I think it's a very important space for functional precision medicine, which is to optimize the current options of treatment that are already in the guidelines.
Is there a chance you could do that with newly diagnosed patients since you are already testing NCCN guidelines, or it's going to be very hard to get
Because the
approval for that?
newly diagnosed, usually they just have one option, the first line. So the first line is very restricted. That's the thing. And so that's why we went to second line and third line where you have options, because the first line usually it's one option and they don't want to move from there.
Yeah, I saw that you're giving the opportunity for the physician to put in what medications they're thinking,
Exactly.
and then you're
And then you
randomizing. And
randomize.
that's a smart way to go about it.
Yes, and I'm forcing them
Yeah.
be the ones to click and to introduce the options, because I didn't want to be the monitors or to be so that the physician really has to think which are the options for that patient, taking in account the toxicities and the patient he has in his at her front, and to see really what could be the options for that patient. And then
Are you able to test different doses? Are you able to test?
no.
No.
Well,
So this
we
is
do, but it doesn't make any sense to do that in the fish. I don't think so, because also these drugs have been, tested in so many trials, and the doses have been tested. So I don't think the fish is good for testing doses. We always use the maximum tolerated dose to the fish to be sure that if we don't have a response, it's not because of problems of dosing. It's just cells are really resistant to the treatment. This was at least was my rationale when designing this.
You're saying maximum tolerated dose for the fish. What about the Cmax?
So we always do the maximum tolerated dose of the fish based on the Cmax of the patient. So we always go from the Cmax. We go into the literature to find the Cmax of that drug, and then we go to the fish and increase the dose and sometimes we are exactly the same as Cmax. Sometimes we go further because the fish can cope with it. Sometimes we have to go lower because it's too toxic for the fish. But most of the time it's okay.
It's too toxic for the fish. Imagine the toxicity.
Yes.
Humans.
Yes.
Thank you. Sorry, I don't want to take most of the time. Anyone else has questions? Feel free to just ask questions. No? Well, I'll ask one more question. At the end, when you're looking at the caspase-3 staining and the immunofluorescence, like how much data do you get? Like you're really you're doing a Z-stack, right?
Yes.
That's a lot of data. How are you managing all that data to be able to put everything together and return data to the oncologist? Just curious.
That's why we are trying to develop a AI program to help us. Yeah, at the moment we are doing that to see.
And how long does it take? I mean, you can still return results in 10 days.
So overall, we do it in 10 days, but we want to reduce it with this AI help. That's our goal. So because
Really cool.
our essay is 3-4 days, then the rest of the days is to do the imaging and analysis.
It's imaging and analysis.
Yeah. So more than half of the time is to do that. So so with the AI we try to reduce that time.
Thank you. I guess there are no questions. So thank you so much, Rita. That was amazing.
Everyone.
All right. Well, we'll talk. We'll see you soon, hopefully. Thank you, everybody.
Bye.
Bye.